Hello everyone, my name is Nathan, and today I'm going to be showing you how to extract DNA from a strawberry. For this experiment, you'll need the following materials: a half cup of water, 2 tablespoons of non-scented soap or shampoo, 1 teaspoon of salt, 1 Ziploc bag, a cup, a spoon or some mixing material, a strainer, some coffee filters, about a half cup of isopropyl alcohol. The higher percentage the better. Here I'm using 91%, and make sure you throw this in the freezer to allow it to chill, as I'm doing right now. And finally, you'll need strawberries. Now, before we get into the experiment, DNA, also known as deoxyribonucleic acid, is the molecule that contains the instructions for an organism, allowing it to reproduce, develop, and live. DNA is inside every cell and passed down through family generations. DNA is made up of nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. There are 4 different nitrogen bases. These are adenine, also known as A, thymine, T, guanine, G, and cytosine, C. These nucleotides form together to make a double helix, which looks like a ladder. The phosphate and sugar molecules are on the sides, and the base would be the nitrogen. Adenine pairs with thymine, and guanine forms with cytosine. This is known as hydrogen bonding. A fun fact is human DNA contains about 3 million base pairs, and of that, 99% of all humans contain the same base pairs. The DNA is then packed very tightly until it comes to chromosome. Each chromosome contains a different set of genes, and the total number varies between different animals and plants. Humans contain 23 pairs of chromosomes and 46 total, which are found inside a cell's nucleus. Strawberries, however, contain 7 sets of chromosomes, but are aukuploid, meaning they contain 8 chromosomes in each set, totaling 56 chromosomes. Now we will begin the experiment. First, we will be making the extraction liquid. Add the water, salt, shampoo or soap in 1 large bowl or cup, and then mix the solution together, and then put it to the side for later. Next, you are going to cut the leaves from all the strawberries. Once you're done with that, add all the strawberries to the Ziploc bag. Now, with your fingers, smash the strawberries. This step allows the strawberries to become structurally damaged, allowing the strawberries to release their DNA. Smash the strawberries for a few minutes until it looks something like this. Now we will add the solution to the plastic bag with the strawberries, and then begin to smash and mix the strawberries for a few minutes once again. This solution is very important for extracting the DNA because the solution helps lyse the strawberry cells. The shampoo is used to help dissolve the cell membrane, and the salt is used to remove proteins bound to the DNA. It also helps allow the proteins from being dissolved from the alcohol. Next, we must strain the mixture 2 times. First through a strainer to get rid of any of the large pieces, and then second through a coffee filter to get rid of the smaller pieces. You will need some patience with the coffee filter, as it does take a while. After straining the solution, there should be no large or small pieces left in the mixture, and now the mixture is ready. Now grab the ice-cold isopropyl alcohol and add it to the mixture. The alcohol allows the DNA to precipitate and allows the DNA to clump up, since the DNA is non-soluble in isopropyl alcohol. The reason we're using ice-cold alcohol is because it allows the DNA to separate from the strawberries faster. Immediately right after adding the isopropyl alcohol, you'll begin to see this white stringy stuff on the top. This is the DNA. Within waiting a few seconds, you'll be able to scoop out the DNA. Make sure you do not mix the solution, as you do not want the strawberry liquid and the alcohol to mix. Once all the DNA rises to the top, you can scoop it out. Here, I'm using a spoon to scoop out all the DNA. Congratulations, as you have now successfully extracted DNA from a strawberry.